Okay, so let's go ahead and continue with part 2 of chapter 16. This is the second part of the host microbe interaction lecture. And before we get into the next couple slides, I just really quick, I added this slide really quick just to explain myself, because I made a couple of really important errors in my little quick off the cuff story about the syphilis study. So one, they were not soldiers. They were sharecroppers. They were poor black men in, I want to say, Tuskegee is actually a city in Alabama. And so when I heard Tuskegee, and also it was carried out by a military, it was called so it's no longer around, it's called the PHS, which is the Public Health Service, I think. At the time, it was a branch of the military. So I got those two things confused. So they weren't, the Army didn't give their soldiers syphilis. That's not what happened. But what did happen is they basically took a cohort of African-American males, and they didn't tell them that they had syphilis. And they basically just tracked their disease progression over 40 years. And it didn't end until the 70s. And there is some controversy here because a lot of the evidence was destroyed over time when the scientists that were at the time carrying it out later because this took place over 40 years. A lot of the evidence and a lot of the data that was collected was destroyed. And a lot of people are very suspicious that the physicians at the time gave the participants syphilis on purpose. And there's no way to prove that or not prove it, but the story, the for sure story, there's no proof of that. So it's important that I don't, say things off the cuff, because of course that's not good science. So if you want to learn more about this, there's lots of articles online. I would encourage you to look through PubMed. If you don't know about PubMed, it's a really great website. It's basically ncbi. I think it's .gov. Yeah, here it is up at the top. You can see the little kind of the shorter video. I also included the citation here if you want to click on it and read the actual article. But it's a really interesting, contextualism is important. And 1940s America was different than 1970s America, and is hopefully different than 2020 America. And I'd like to think that we're getting better and that something like this wouldn't happen today. Okay, so let's continue on with the rest of the lecture. Okay, so we're talking again all about how. So at this point, let's put this in context, right? We have all of these bacteria in the world. The vast majority of those bacteria want nothing to do with us. They don't like humans. They don't want to be on humans. They don't want to be around humans. Not the right temperature, not the right oxygen, not the right anything. Now, there's a small subset of those bacteria that actually like living in us and on us, and the vast majority of them actually have a good relationship with us. We get along with them, they colonize us, and they sometimes give us things, they give us advantages, they keep out the bad guys, and so on. But every once in a while, we'll have a bad actor, right? We'll have a bacteria that represents a proper pathogenic threat to our health. And that's when our immune system kicks in and we have physical barriers against that infection and so on. So let's talk about some of the tricks that these pathogenic bacteria use to get into the host or attach to the host and so on. Remember that bacteria have to, if they don't figure out a way to grab onto something, they're going to get trapped up in the mucosa and swallowed or sneezed out or otherwise destroyed. They don't want to get caught in the slime trap. They want to attach to an epithelial cell so that they can hang on. And so bacteria that have these, what they're called adhesins, and they're usually on the end of their pili. Remember those pili are those little hair-like extensions on bacteria. And at the end of those pili, they have these adhesins. And what's really cool about these adhesins is that they're actually tissue specific. So I think the example that they give in the book is Neisseria gonorrhea has a specific adhesion that makes it uropathogenic. In other words, it can hang on to specific tissues in our urogenital tract. So the type of pili and the type of adhesins that are attached to that pili can be considered a pathogenicity factor for bacteria. In other words, it allows them to grab on and hang on to these epithelial tissues, whereas other bacteria might get washed away by our mucosa. So our host, of course, we have a couple of ways to protect ourselves from that. One, hopefully we already have other bacteria there that are crowding them out and not letting them get close to that epithelial surface. And then we also have sticky traps in our mucus. Remember that we have that S, I think here stands for, what does the S stand for? Secreted? Soluble? Again, I should know this. What the heck is wrong with my brain today? I want to look that up because it's going to bug me. SIGA. That's probably going to come up with something really weird, of course. IGA. Will that do it different? It figured. How about alpha? Let's try alpha. I probably should pause the video at this point. All right, IgA. Of course, the food chain. IgA. IgA, immunoglobulin. Oh, secreted. I think I just saw it right there. Yes, yeah. Secret. Yeah, that's what I thought. Secreted form. So basically, in the mucosa, which is its predominant form, right? So remember that in the serum, in our blood serum, in our blood, the predominant form of antibody is IgG. In our mucosal lining, it's IgA. So IgA is going to grab onto those bacteria. And many of our IgA, remember that antibodies are specific, and many of the antibodies, our IgA antibodies that are in our mucosa, are specific for these adhesins. So basically they grab onto these adhesins, and if they do that, then the adhesins can't connect to the receptor, and it doesn't allow the bacteria to connect with that receptor. All right, so again, the vast majority of time, if we have an intact, healthy mucosal layer and an intact, healthy skin, bacteria will not get in us. It's our best, by far, our best defense against invasion and infection. And the only way that most bacteria can get in us is through passive penetration. In other words, if we have some type of abrasion or some type of compromise to that layer. So if we get wounded, if we have a scrape, a cut, a burn, and then one that people commonly overlook is a bug bite, right? So when we get bit by a mosquito or a tick or whatever it might be, that's going to compromise our physical barrier and allow bacteria or other things to get in. It's important to also understand that, just because you don't see an abrasion or just because you don't notice a compromise of our system, it doesn't mean our barrier, it doesn't mean that it's not there. So for example, a really unfortunately, a really common source of this compromised barrier is gum disease. So, so many people have gum disease because we don't floss and we don't take proper care of our teeth, and we have really poor dental care in general in the United States because many people don't even have dental insurance. Because we have that compromised basically interface with those bacteria can get inside that compromised barrier, for example. Major source of problems, and there's some intriguing evidence that's pointing to chronic disease and gum disease being connected somehow. All right, so other bacteria can secrete enzymes that somehow modify the host receptors or the host cell itself. And some of those enzymes can also cause endocytosis. So there's these really cool secretion systems in gram-negative bacteria. So here we have the bacteria up here. We have a host cell down here. And the bacteria actually creates this like syringe-like puncture weapon. Basically, this looks like alien stuff, right? And it punctures the host cell, and it's going to secrete some type of effector molecule into that cell. Sometimes it's an enzyme, sometimes it's a smaller molecule, and it's going to modify that host cell somehow to allow, sometimes like it induces phagocytosis, like that bacteria is an intracellular pathogen, it actually wants to be inside that cell, but it modifies the host. So there's all kinds of really cool tricks that bacteria can come up with to get inside the host and cause, basically establish an infection within the host. So I mentioned that some of the bacteria want to, I shouldn't have said phagocytosis, I should have said endocytosis, right? So because phagocytosis means eating and implies destruction, and what I should have said was endocytosis. So the cell takes in that bacteria, and for various reasons that bacteria can survive within that cell, so inside that cell. And here's some examples. Chlamydia is a perfect example. It's an obligate intracellular bacteria, and that's one of the reasons why our body has a difficult time fighting it off, right? I mean, it's fairly easy, my understanding anyway. I of course don't know personally. But my understanding is that antibiotics take care of chlamydia pretty easily. But you need that antibiotic. I think it's a shot that you have to take, and then you can clear it pretty easy. But otherwise, it's really difficult for your body to clear because the bacteria is hiding inside a cell, and that's more difficult for our immune system to handle. And would it be? Ooh, here's a good question. Would it be humoral, cell-mediated, innate? What branch of our immune system would take care of intracellular pathogens, so bacteria that hide inside our cells? Well, it would be cell-mediated, right? It would be those cytotoxic T cells, because hopefully those cells are going to process that bacteria or take some piece of that bacteria and display it in the MHC1, that badge, remember that security badge, and they're going to display it on the outside of the cell so that our cytotoxic T cell can recognize it and kill that cell. Right? Our cells kill themselves all the time, right? They want that. They will gladly sacrifice themselves for the host, for the greater good of the host. Our cells are socialist. Didn't you know that? We're all socialists. Okay. All right. So other microbial virulence factors. So we've of course talked about capsules endlessly, right? Without a capsule, bacteria doesn't stand a chance. And capsules are really good. They do a good job of basically denying those phagocytic cells from engulfing the bacteria, and they also can help them grab onto things. And they also, don't forget, make them, how would I say this? They make them less antigenic. Right, so the capsule, remember, is like a slime layer. And so if you think a slime layer like that silly putty, right, it's hiding all of those antigenic markers on the surface of that bacteria, so our immune system can't find them as well. So our those antibodies that are trying to attach to that specific epitope can't get to it because there's that mushy slime layer preventing the antibodies from attaching. Other bacteria have some pretty interesting tricks that they can pull. So it's not just a slime layer that they use. Some bacteria secrete little peptidases. So they're enzymes that can cleave proteins. A peptidase is basically an enzyme that can destroy specific proteins. And these enzymes will destroy complement. Remember that, is it C3b? I think it's C3b will attach to bacteria, and that opsonizes the bacteria and allows those phagocytes to eat it easier. And so as soon as that complement lands on the bacteria, that peptidase will dissolve that complement, so it doesn't work. Pretty cool. It's kind of like, it's almost like an arms race. Our body has something that can do this, while the bacteria is going to evolve to fight that, and so on. And then the FC receptor, oh goodness, I should have put a picture in here. Let me find the picture in the book. It's actually pretty cool. Maybe I can draw it too. It's in chapter 16. What? It's figure 1610. 1610 in your book that will draw a better picture than I will. But if this is a bacteria, remember that our antibodies look like Y. They have the kind of the Y shape, and they're attaching, that antibody is recognizing specific epitopes on our bacteria. So some bacteria, this is I think this is kind of ingenious if you ask me, but remember, bacteria aren't smart. They don't have a brain. Anyway, what they do is they'll have a specific receptor on the outside of bacteria that basically that recognizes the FC portion of our antibody. So they basically force the antibody to have the wrong end sticking out. And remember that the FC portion, right, of an antibody, this is the FC portion. That's the portion that kind of amplifies that signal, and that's the portion that we need to communicate with other aspects of our immune system. So if that portion is stuck to the bacteria, it can't do anything. I just think that's such a cool little trick that bacteria do. The other virulence factor or pathogenicity factor that I want to talk about are toxins. And so we have two categories of toxins, exotoxins and endotoxins. And I do want to spend a little bit of time on these because there's something that they are a topic that's important to you as health professionals. So let's talk about exotoxins first. Exotoxins are probably what you think of when you think of a toxin that's produced by a bacteria. So again, we're not talking about viruses at this point. They don't make any toxins. It's just bacteria. And I can't think of an example where it's not a protein. They're almost always proteins. but so because they're proteins, they're very strong antigens. So our immune system can recognize them quite easily. The problem is that they work super fast, and you need a really, really small amount of them to be lethal. So by the time our body can mount an effective antibody response, because it's strongly antigenic, we're already dead. So our body really doesn't have time to kind of go through the clonal deletion, clonal selection, and T helper cell development and production of plasma cells and all of that, because the toxin acts really fast. The good news is that they're heat labile, which basically means we can destroy them quite easily by boiling. Basically heat sterilization. If we autoclave something or boil it, we will sterilize it and remove any of those exotoxins, because remember that proteins get denatured quite easily by high temperatures. Exotoxins have a whole class that's associated with them. So exotoxins that destroy or somehow damage nerve cells, for example, are called neurotoxins. Toxins that cause problems in our gut are called enterotoxins. Leukocidins specifically target immune cells. Hemolysins destroy red blood cells. I think there's a general one called cytolytic or cyto. I feel like it's one of the words that I think I crossed it out actually, but they're cytotoxic, so they're basically generally toxic to cells and are harmful to cells of our body and so on. So we have all these different effects and that these toxins can have depending on the toxin. I just want to point out two of them that you've probably heard about, of course, and they happen to be members of the Clostridium family. I feel like we worked with one of a Clostridium species at some point in lab, but Clostridium species tend to be anaerobic. In fact, I think they're obligate anaerobes. But Clostridium tetani, of course, is the causative agent of tetanus. And so look at that LD50 for a mouse. It's 4 × 10 to the -5 micrograms, right? A microgram is smaller than you could possibly imagine, and it's 0.00004 micrograms that would be considered toxic. The smallest amount of this toxin is a lethal in amount, and it's not that much more for people. I mean, I think that there's some I'm going to screw this up, I think, but I want to say I was watching a video the other day that said that 500 grams of tetanus toxin it was either tetanus or botulinum toxin can kill the entire population of the United States. It's extremely small amount, because we're talking about proteins that are just so incredibly tiny. And of course, we've heard of botulinum toxin, known as its much friendlier name of Botox. So Clostridium botulinum, this is, so historically, we've known it as the bane of home canners. I don't know if that's even the right word to use, but people that can things at home, right? So if you have a garden and you cook a lot and you put food up, so you make jam or you make pickles or something, and you don't sterilize it properly, in other words, you don't bring it up to a certain temperature, what happens is, one, you kill the vast majority of bacteria that are in there, and you're also creating an anaerobic environment. So you're creating the perfect I'm forgetting a huge part of this. The Clostridium species, their big survival factor is that they form spores. And if you recall from way early in the course, spores are really resistant to heat. So you have to have that autoclave level of heat. I think it's 120 Celsius for at a certain PSI for 20 minutes. And if you don't do that, those spores can survive. So if you have, and they're in the soil, right? Clostridium botulinum, Clostridium tetani are in the soil. If we went to I guess anywhere that has like your typical soil microbial population, you're going to find these two bacteria in there, or at the very least spores. And so if you think about having a garden, right, and you're not processing those vegetables correctly, it's possible that you could get a spore from botulinum in your canned goods. You don't sterilize it properly, and now that spore has like the perfect environment in which to grow. And so botulism was long associated with canned food items and was extremely lethal. And it's not so much anymore, mostly because people don't do it anymore. Right? Very few people actually can home food items. And when they do, we have better pressure cookers and better canning systems and so on. So it's not as dangerous as it used to be. And of course, now most people associate this bacteria with Botox. And when Botox first came out, it was all about wrinkles and getting injections into your face. But it's turning out that that Botox toxin has a lot of other effects. For example, you can inject it into your armpits and you won't sweat. So you're basically kind of causing paralysis of the nerves that cause you to sweat in your armpits. It also, there's some promising research being done on migraine. So you can get botulism injections, I should say into your head. Saying into your head sounds like it's going into your brain, but in specific kind of what would that call it? Subdural, right? Like under the skin in specific areas of your head, and it can help with migraines too. So there's some interesting research going on with the Botox, the actual toxin itself, because of its ability to essentially make a nerve stop working. Okay. Is that oh no. I think I accidentally deleted a slide that had the AB toxin. I'm a mess. AB. Well, guess what's not on the test? AB toxins. And they're kind of cool. They're toxins that have two parts to them, and they're a fun area of research, so I always try to mention them. But I'll just take that. If I have a question on that on the test, I'll take it out and I will let's just go back. I'll do this now while you're watching so that you believe me. Let me go back to the learning objectives. I want to say that there was something on AB toxins. Yeah, outline the mechanism of action. Unfortunately, we're gonna have to take that off the test. How did I delete that slide? That is just weird. Okay, let's talk about endotoxins. Okay, so endotoxins, also a toxin, but very different than exotoxins. So let's talk about, I'll describe what endotoxins are first, and then we'll talk about all the ways that they're different. So endotoxins, despite the term endo means inside, and it's kind of, unfortunately, a misnomer, because it's really on the outside, specifically of gram-negative cells, and it's actually LPS. So remember that LPS is part of that outer membrane of gram-negative cells, and there's two parts to LPS. There's the lipid A portion and the O polysaccharide. And because they're a lipid is very poorly antigenic, and the polysaccharide, the O part, can vary quite a bit. Our body can't make a good antigen to this. So LPS has always tricked our adaptive immune system, and we just don't make good antibodies against this. It is, however, a big red flag to our innate immune system. Remember that those toll-like receptors on what our dendritic cells and our neutrophils and our macrophages know what LPS is. It's an ancient enemy of chordates, and well, actually of all multicellular organisms. So our adaptive immune system does not work well against this endotoxin. Oh crap, I forgot to I have to go back and explain toxoids. Our adaptive immune system does not recognize LPS very well, but our innate immune system does. And here's the other problem with endotoxins, is that they can actually withstand autoclaving. So we can't just autoclave something and heat it up and destroy this endotoxin. However, it is fairly rare to get it from, say, like a contaminated scalpel or something like this. What typically happens is we get sepsis. So if we have a gram-negative infection that has somehow taken hold within our body, and we enter into a state of sepsis or bacteremia, where this this gram-negative bacteria is in our blood, and then we treat that disease, what's going to happen is the act of destroying that gram-negative bacteria is going to allow that LPS to get basically get loose and come free of the membrane. And that's when it causes the problem. So oddly enough, an intact gram-negative bacteria isn't a problem. It's the dead bacteria that releases the LPS from its outer membrane that causes the problem. So we have all this bacteria in our body. It starts to get destroyed by either our immune response or an antibiotic that we've been given. And now suddenly our system is flooded with LPS. And what's going to happen is it's going to piss those neutrophils off, right? Remember, those are the guys that throw the hand grenades. It's going to turn on our toll-like receptors. Our macrophages are going to go into action. And basically our body goes into what's called a cytokine storm. And we have inflammation and anger throughout our body at the molecular level. And this can cause coagulation throughout our body. It's called disseminated intravascular coagulation, and we enter into a state of, it's almost like toxic shock syndrome, and it's deadly. It's a very big problem in treating these gram-negative infections because you can enter this kind of state of shock very quickly, and it takes people very, very fast. The difference between, so there's lots of differences between endotoxins and exotoxins. So again, if we, like, I can imagine we had a scrape on our knee and we got an infection with a gram-negative bacteria, it's going to be, it's going to be localized to that. This really has to be a systemic infection. So we need a really large amount of this LPS to cause an effect. And that's, of course, in contrast to those exotoxins, which we need just mini micro levels of that protein. So the good thing about endotoxins is that we need a lot of them. We need to have a big effect to have really deleterious effects on our body. But of course, the problem is that we can't make an antigen, or we can't make an antibody to it, and we can't get rid of it by autoclaving. The other big difference between an endotoxin and an exotoxin that might not be obvious in this talk is that there are hundreds that we know about. There's probably thousands of exotoxins. There's lots and lots and lots of exotoxins, basically proteins that are secreted by bacteria that cause us problems. There's only one endotoxin that we know of, and that's LPS. So lots of different kinds of categories and specific types of toxins that are exotoxins, but only LPS is considered an endotoxin. And I wanted to go back because I realized I didn't talk about what a toxoid was. Right, so let's talk about this middle bullet here. So because these, if you remember, I talked about how they're so fast acting, and they usually act before our adaptive immune response has the ability to create antibodies. So this is one of those things for which vaccination is so important. For example, we all get our tetanus booster every so often, right? So how does that tetanus booster work? Well, we'll talk a little bit more about this in Unit 5. But what we can do is we can take that toxin and destroy it using heat. And now it's considered a toxoid. And now that toxoid can be considered a vaccine. So we introduce that toxoid to our body. Our body mounts a small immune response to it, and it now recognizes that protein for the future. So this is the way that we're vaccinated against toxin production by, in this case, Clostridium tetani. So vaccinations are one way of avoiding the toxins. I think there might be I feel like there might be a vaccine against botulism, but I feel like it's such a low risk that it's not like it's not really a viable commercial product. I'll have to look that up. So then the other thing is, like, let's say that we think we have tetanus, right? Like we never got vaccinated for it. We step on a rusty nail and things are going south fast, right? First, you need to get to a hospital. But what can happen is they can actually start you on a drip of antitoxins. So they basically give you purified antibodies. It's a commercial product, and those antibodies will go into your bloodstream and neutralize that toxin. Because remember that antibodies can be specific for these proteins. And so a group of one class of antibodies that we talked about in the last chapter were called antitoxins. Cool. So exotoxins, another difference between exotoxins and endotoxins. We can make antibodies against exotoxins. We can't make antibodies against endotoxins. All right, so again, if we were in class, we'd kind of take a few minutes to talk about this. I already did kind of address several of the major differences. There's also a nice table on page 431 of your text. It's table 16.2 that has a nice compare contrast between these two exo and endotoxins. Okay, so very briefly, I want to talk about the progress of a disease. So we're in the bad area of host microbe relationships. We're talking about a disease state caused by an infection. And there are different, in fact, I want to kind of get rid of prodromal because it's not really useful for us. But I want to talk a little bit about how acute diseases progress. Remember there's different categories of disease. So we're going to talk very briefly about acute diseases. And this is usually what you think of when you think of disease. But remember that you can have a chronic disease or a latent disease as well. So a chronic disease would be something like hepatitis, where once you get it, you have it for a very long time and you have to manage it for the rest of your life. Whereas on the other hand, you can also have a latent disease. Perfect example is chickenpox, right? We get it, there's an incubation period, illness, we get over it, but it stays in our body and can come back. And when it comes back as syphilis. Oh my goodness. Syphilis. You don't get syphilis from chickenpox. You get, shingles. It comes back as shingle. Oh my gosh. Oh, don't tell anyone I said that. So we have latent diseases. So I don't want to address chronic or latent diseases. I just want to very briefly talk about acute diseases, which is the common, especially for bacteria, it's really the common route or the common progression of a disease. So we get a disease, we have an incubation period, and it's the time between which we are infected and we start to show signs and symptoms. And this can vary. And we're seeing that now, right? I read a really great article the other day about the difference between COVID-19 and SARS. They're both coronaviruses, but the reason is that, so the SARS virus was a big deal, and it was actually much more deadly in terms of virulence than the COVID-19 virus. The issue, of course, is that when people got SARS, they got it immediately, and they immediately started showing signs and symptoms. They started showing signs and having symptoms. Whereas this COVID-19 is such a booger because there's a huge incubation period. Basically, we can get it and we don't notice it until up to 14 days, sometimes even longer from when we get it. So there's a lot of talk about having these asymptomatic carriers of the virus that are walking around and infecting other people. So we have this incubation period, and then once we start to show signs and have symptoms, we can then, of course, either A, die, or B, recover. And that period of convalescence is where we have decline, and that's kind of the bridge between illness and convalescence, and we have a recovery to a pre-disease state. So acute disease progression is again what we commonly think about when we think of infectious disease. The pathogen establishes infection within the host. It establishes illness. We get over it and we convalesce. Okay, so briefly again, there's only a couple questions that are pretty straightforward on the test on this last bit. And again, I think this is section 19.7. Why does it say 1914? Let me see what figure that is. I don't think that sounds right to me. 1914. 1903. That's what I thought. That didn't sound right to me. 19.3, 19.2. Sorry, hold on. 19.14. I wonder if that has to be a mistake. So, sorry, let's go ahead and cross this out. I'm not sure what that's on. But I wanted to talk just very briefly about, you can't talk about host microbe without the idea that when you're in a hospital, if you work in a hospital, and we're seeing that, of course, better than ever, what a better time to talk about this, but when you work in a hospital, when you are staying in a hospital as a patient, you have an increased risk of what's called nosocomial infections, or HAI, healthcare-associated infections. And I should just say hospital, of course, we're seeing this big time with nursing homes and hospice care and assisted living facilities and so on. So we can have, the definition of a nosocomial infection is something that has been acquired as a result of being in a healthcare facility. And a fairly large amount of patients, 15%, up to 15% of patients can get these nosocomial infections. And it happens for a lot of reasons. And hospitals are a lot more dangerous than people might think. You think of that they're very clean and they're sanitized and they usually have like white walls everywhere. But one, if you're in a hospital, chances are you're compromised somehow, right? You have a compromised immune system, and just the stress itself of being in a hospital could lead to a compromised immune system. Also, there's other sick people there. So there's a huge reservoir of these organisms in the hospital itself. And of course, we're seeing that too, right? People are saying, unless you absolutely have to go to the hospital, don't go, because just stepping in a hospital right now is putting you at increased risk of getting, in this case, of course, COVID-19. So there's some, this is just anecdotally, I was talking to a friend of mine that works in a hospital, and she was saying that basically they're doing, she lives in and works in South Bend, Indiana, and she was saying that they're doing stuff right now with PPE that they would normally get fired for. They'd get written up and fired for, and like, for example, reusing face masks, reusing gloves, not changing their apron before working from one patient to the next, and so on. And they're of course reusing it because they don't have any more, right? All these hospitals are dealing with shortage of PPE, and so this is putting these patients at an increased risk of getting in this case this virus. So where do patients, so this actually addresses two things. So what are the most common routes of transmission? This is the bottom pie chart. And what are the most common agents? And you can see, let's start with the agents, because this pie chart basically tells you that it's a smorgasbord, right? There's so many different pathogens that can be the causative agent. But if you notice, here's our three big ones. So basically three species, or at least Streptococcus is a family, or genus, I should say. These three make up about half of all infections. E. coli would be your gastroenteritis, basically a gut infection. Staph aureus is going to most likely be from a compromised skin, bed sores, for example, and Streptococcus, the same thing. It's going to be a route of entry through your skin. So those three bacteria alone make up about half of those nosocomial infections. But how do they get in? Well, again, here, a little bit less diversified. So bacteremia, of course, means introduction directly into the blood. Urinary tract infection because of catheter use. Respiratory tract infection, which is kind of interesting that it's that high, to be honest. I'm surprised that it's really as high as 15%. And then infection of the wound itself. So others could be like at the port, at the site of insertion of a port or some other medical device. What are some other possibilities? I'm trying to think what else. I feel like there's another comment, like I guess like skin grafts and burn victims would be considered part of the surgical site. That's a big one. But again, it's keeping that skin intact and keeping your epithelial mucosal layers healthy, and that's going to prevent most of those disease transmissions. I think that's the last slide. Yay, it is. And I'm a little under 40 minutes. Not too bad. Good. I hope you enjoyed this host microbe. I enjoyed talking about it. It's kind of a difficult chapter to because it's a lot of concepts. It's hard to find good questions to ask students that press their knowledge on it. But it's a really important conceptual chapter to think about for all of you, because you're going into the healthcare field and you need to think about what is our relationship with bacteria. Of course, now the conversation so much is about viruses, but way more people die of bacterial infections, and I shouldn't say die, but go to the hospital because of bacterial infections than viral infections. Far more common. So the relationship that we have with bacteria, again, has always historically been thought of as a combative relationship and a negative relationship, but I want you to hopefully kind of rethink that a little bit and think about what role bacteria play in both health and in disease. All right, I'm done.